Hey, I just want to study. This is like for personal purposes. I'm not sure if I'm gonna keep this public or not, but whatever. So I have an exam coming up. It's about cellular respiration, and I just want to explain it in a way to myself that I want to know in the future. So yeah, let's get started. We're gonna go with the overview of cellular respiration. So this is what an organism obtain energy in a process called cellular respiration. During the bonds of food and oxygen molecules are broken and energy transfer from one interacting molecule to another. New compounds of ATP that form can transport energy into muscles, and the energy needed to maintain body temperature despite ongoing energy transfer to the environment is released. And here's the summarized picture of it here. It's going to happen in the mitochondrion, and there's a few steps that I want to explain first of all. So as you see in this photo, there's something called a glycolysis. The end is referred to as breaking down, and the glycol is considered as glucose. So what it means here, you're breaking down glucose and you're gonna make it small enough to enter the mitochondrion, because if it's as glucose it cannot enter it because it's too big of a molecule. So to do that, you're gonna have to break it down into two pyruvate molecules. Is pyruvate even a molecule? Okay, these are the weak questions I don't need to start asking myself now. Okay, so this happens before we begin. So this is in the cytoplasm, also known as the liquid of the cell. And here what it's gonna do, it's gonna turn the formula C6H12O6, and it's gonna take that 6 carbon and it's gonna divide it into two. So it's gonna be 3 carbon, 3 carbon, and that's gonna be the two pyruvates. So that generates ATP, in which we're gonna use later on in the electron transport chain, or also like chemiosmosis. And when it turns into the pyruvate, it's gonna be able to enter the mitochondrion. So that's when it comes to the preparatory reaction, which I'm hopefully gonna explain to you in a bit. Also, like a quick note, there's the Krebs cycle, and as I remember, because we have two pyruvates, the Krebs cycle is gonna turn 2 times because we have two of the pyruvates, not simply one. Okay, let's keep going. Yeah, as you see here, this is the glucose, the C6H12O6, that's the glucose. And cellular respiration has two main parts: the glycolysis, and it's an anaerobic process. An anaerobic process does not require oxygen, but an aerobic respiration includes Krebs cycle and electron transport, and that's aerobic, so you need oxygen to be able to complete those. So we'll get to glycolysis now. Glucose, as I said here, is broken down the cytoplasm through a process of glycolysis, and 2 molecules of ATP and 2 molecules of NADH are formed with each molecule of glucose that's broken down. And in this figure 11, we're gonna follow as we read about the steps. First, we have 2 phosphate groups that derive from the 2 molecules of adenosine triphosphate are joined into glucose. And we notice that some of the energy to ATP is required to start the reaction to produce the energy for the cell. And as you saw here earlier, the 6 carbon molecules are broken down to 2 carbons, so it's 3 carbon compounds, and the 2 phosphates are added, and electrons and hydrogen ions combine to 2 NAD plus molecules to form 2 NADH molecules. So a little note here, the electron carriers that we're going to have is NADH and FADH2. Okay? An NAD plus is similar to NADP, which is the electron carrier in photosynthesis. And last, the 2 3-carbon compounds are converted to molecules of pyruvate. At the same time, 4 molecules of ATP are produced. Yeah, so that's what you got here. Here's the little photo if you want to look at it for a moment. Yeah. So now we get to the Krebs cycle. And the glycolysis has a net result of 2 ATP and 2 pyruvate molecules. Most of the energy from the glucose is still contained in the pyruvate. And in the presence of oxygen, pyruvate is transported into mitochondrial matrix, where it eventually converts into carbon dioxide. The series of reaction in which pyruvate is broken down to carbon dioxide is the Krebs cycle. And by the way, as I remember, in the Krebs cycle, you're gonna keep breaking down the CO3, and you're gonna create a lot and release a lot of CO2. Okay. And as I mentioned earlier, the word origin of glycolysis, the lysis in the end is to rupture or break, and the glucose is like the glucose itself, so you're breaking down glucose. So now for the preparatory reaction, just a little note here, because we just want to reshape the pyruvate, so it's gonna turn that carbon, the 3 carbon, and it's just gonna reshape it a bit until it looks like an acetyl CoA. So prior to the Krebs cycle, this is the preparatory reaction now. The 3 carbon need to react with the coenzyme A or CoA to form 2 carbon intermediate called acetyl CoA and carbon dioxide. So these 2 are gonna be formed, and honestly, it's not really something changing. It's simply that we're gonna turn the pyruvate into an acetyl CoA. It's just like you light a candle, and just because you want to change how the wax is shaped, you just light it up and you change its shape and it stays the same, you know. That's kind of the goal for it here. So NAD plus is converted to NADH. Carbon dioxide is given off by the cell. Since there are there's 2 molecules of pyruvate for every preparatory reaction, it results in the production of 2 carbon dioxide molecules and 2 NADH molecules. And then we move the acetyl CoA to the mitochondrial matrix. So here, I don't know if I really mentioned this earlier or not, but since we have 2 pyruvate, we're gonna have the citric acid cycle turn twice. But like, I'm still unsure if this is true or not. I just remember learning that. So now we go to the Krebs cycle, and we're gonna read it out loud here. The Krebs cycle begins with acetyl CoA combining with a 4 carbon compound to form a 6 carbon compound known as citric acid. Do we get it here? Or are we still confused? Okay, I'm actually kind of curious in this part. We're just gonna research it, even though I do know it. My bad if the keyboard sounds like a bit annoying. Oh, there's Quizlet and Lumen Learning. Let's try this. Okay. Oh! Okay, well, this is a bit complex to me. Okay, yeah. Okay. Oh, okay. Multiple theories can be used. We're going to continue note cider as aerobic catabolic state that needs energy. Under these conditions, acetyl CoA will enter the citric acid cycle known as Krebs cycle or TCA cycle. And it shows it. The citric cycle begins with acetyl CoA or 2 carbons combining with 4 carbons to make citric acid or 6 carbons. Okay, a series of transformations occur and is giving carbon dioxide and NADH is produced. This leaves alpha ketoglutarate, which is 5 carbons. Okay, I think I'm gonna go as it is for the flow now. So the citric acid is then broken down in the next series of steps, releasing 2 molecules of carbon dioxide and generating 1 ATP, 3 NADH, and 1 FADH2. As I mentioned, NADH and FADH2 are going to be our electron carriers. FAD is another electron carrier similar to NAD plus and NADP plus. And then acetyl CoA and citric acid are generated and the cycle continues. So let's look here. Pyruvate from the glycolysis. CO2, the NAD plus turns into NADH. Here's the acetyl-CoA there. And then that's where the citric acid begins. And the 5 carbon molecules. Oh yeah, that is a lot of formations here. Wow. Okay. And just a note here that after the citric and acid cycle, you're going to release a lot of carbon dioxide, like a lot of them, because that's the goal, is to keep breaking down the carbon trioxide, you know. Okay, so let's recall that the 2 molecules of pyruvate are formed during glycolysis, resulting in 2 turns of the Krebs cycle for each glucose molecule, as I told you. The net yield from the Krebs cycle is a 6 carbon dioxide molecule, 2 ATP, H NADH, and 2 FADH2. 10 NADH and 2 NADH2 move on to play a significant role in the next stage of aerobic respiration. Okay, now I'm just gonna sit down here before we start to the next part, and we're just gonna like think and process all that we did. Okay. Just to give a little recap, this is gonna be by voice, so it's you should take a paper and start writing notes down. On the top, write a header as glucose, like in the middle of the page, and put a arrow down and write glycolysis, and next to it write C6 as carbon and 6, the tiny 6, and down there put a tiny arrow and write 2 pyruvate, and next to it write 2 times C3, so it's like 3 carbons, you know. So here we already mentioned how it's breaking down and it has the 2 pyruvates, and you're gonna put 2 arrows. One is gonna be for aerobic, one is gonna be for fermentation. So for the aerobic respiration is the eukaryotic, or the one in the mitochondria, which is the one where oxygen is going to be present. It's going to happen in the Krebs and citric acid cycle, and it's also going to happen in the electron transport chain. So you can write that down in the aerobic respiration. On the second arrow, where the fermentation is, make 2 arrows and write that is going to create lactic acid, and on the second one is gonna create alcohol and carbon dioxide. Yeah, this happens in the... What does this happen in? I cannot even read my handwriting. Okay, wow. It looks like enzymes, but I don't remember writing a Z. Okay, you know what? We're just gonna ignore that last part what I just said. But like the lactic acid, alcohol, and carbon dioxide, okay, yeah, you need to keep that there. So the fermentation, by the way, is the prokaryotic one, which happens in the cytoplasm, and it can happen, or it can be formed without the use of oxygen. That's just a note here. Okay. So we got this. Yeah, we probably got this. Okay. By the way, in the end, where it's gonna be the... where do I call it? Okay, we're not gonna go through here part. Like, not yet. We're gonna go back to where I showed you the electron transport chain and chemiosmosis. So you're gonna have the ETC, also electron transport chain, and chemiosmosis. By the way, if they're gonna ask you what ETC is, ETC is oxidative phosphorylation. This is the ETC. And this is what I remember it being, so if I'm wrong, please correct me on that. And now I think we should start going. And also a really weird note to keep, but always know that organisms, organelles, and cells are all different things. I know you're gonna say that's kind of weird, that you might not get the difference between them, and you might like say, yeah, I do know that, but in the end you might get a mistake off of it. Okay. So to be clear, organisms is something like plants, but plants? It's like plants, animals, etc. Like, just to give a little image for you to think about. So this is organisms, okay? This is organisms. And then organelles is like mitochondria. This is like an organelle, like the chloroplast, mitochondria, all of these. They're known as organelles. Okay, my bad for the keyboard noises. And now the cells is like eukaryotic cells and prokaryotic cells. These are the ones that are cells, okay? So don't go saying that eukaryotic cells are organisms. I'm not gonna try to foreshadow anything, but I think you can imagine from here. Okay, now here's figure 13. And now this is the electron transport chain. The electrons move along the mitochondrial membrane from one protein to another. They start off the electron transport chain begins with 10 NADH molecules and the 2 FADH2 molecules releasing electrons. The released electrons move along the mitochondrial membrane from one protein to another. Here, I think, is it the inner mitochondrial membrane? Yes, okay. The energy carriers are converted to NAD plus and FAD, and H plus ions are released into the mitochondrial matrix. By the way, the H plus ions are pumped into the intermembrane space through the proteins in the inner mitochondrial membrane. This creates an increase in the concentration of H plus ions of the intermembrane space. Why am I saying intermediate? Okay. As a result of high concentration in the intermembrane, here's the inner membrane, like you see that. This is where the E is gonna go, and that chain, the long chain of green, that's where the electrons are gonna go. And those orange ones is where the hydrogens is gonna be pumped out, you know? So there's the intermembrane space where the hydrogens get pumped out, and where they were originally, it was in the mitochondrial matrix, okay? So as a result of high concentration in the intermembrane space and a low concentration in the mitochondrial matrix, the H plus ions diffuse into their concentration gradient back across the inner mitochondrial membrane and into the mitochondrial matrix through the ATP synthase proteins and chemiosmosis. As the H plus ions move through the ATP synthase, ADP is converted into ATP in the mitochondrial matrix. So look, this is just like a scale. And when the scale gets too much on one side, it needs to start and equalize itself. So you see there's a shift, like one has more than the other. So the H plus ions, they go to this, what you call an ATP synthase. Okay, no funny jokes here. Okay, that's not funny even. They go through the ATP synthase and they go inside back into the mitochondrial matrix. Okay, we got that part right. Okay, and oxygen is the final electron acceptor in the electron transport system. In the cellular respiration, the H plus ions and electrons are transferred to oxygen to form water. So you see when they come back in here, the oxygen is gonna accept both the electrons and the hydrogens. You see here, like, you see those little blue things in the bottom where it's next to the ATP synthase, the electron, and you see the half oxygen and the 2H plus. So that, you're gonna mix it together, is gonna make water. So after all the imbalance and all the transport and the electron transport chain, it's gonna form water. We got that part. So the overall electron transport molecules produces 32 ATP. Each NADH molecule produces 3 ATP, and each group of 3 FADH2 produces 2 ATP. In eukaryotes, one molecule of glucose yields 36 ATP under ideal conditions. So it's around 36 to 38, as I remember in my memory. Yeah. I just mentioned it. It's actually in the paragraph below it. Okay. So the prokaryotic cellular respiration, some prokaryotics also undergo aerobic respiration. Because prokaryotics do not have mitochondria, there are differences in the process. The main difference involves using a prokaryotic cellular membrane as the location of electron transport. In eukaryotic, pyruvate is transported into the mitochondria. In prokaryotic, this movement is unnecessary, saving the prokaryotic cell 2 ATP and increasing the net total of ATP produced to be 38. Do we get that? I hope we get that. Okay, now we're gonna have to go through this. Okay. And before we go that, remember that one drawing I told you to draw? So by the way, fermentation, the one that's anaerobic, that doesn't need oxygen, you can actually find that in cramps. Because when you have cramps, one of the reasons is that there's not enough oxygen flowing through that part of the body. And through that you have, and it's the fermentation produces like the alcohol and carbon dioxide, and there's also the lactic acid and whatever, so it's gonna make a burning sensation that you cannot continue because it hurts too much. And the thing is, there's also no oxygen there to keep flowing, and there's not enough oxygen actually, so it's gonna hurt a lot. So yeah, okay, now to the anaerobic respiration. This might seem like I'm speaking to myself again. Okay, so we only have these few ideas. Oh yeah, okay, now we come with other notes. Okay. Oh yeah, did I forget to mention that all the way here, when we're gonna do the Krebs cycle, when the 2-pyruvate turns into acetyl-CoA, it's also gonna be 2 acetyl-CoA and not just 1. Or is that just common sense? We're just gonna consider it like, yeah. Oh yeah, also by the way here, where did we go? When we talked about the cellular respiration here, about the ATP produced being 36 to 38, this is per 1 glucose. It's not for both of them. You don't divide the 36 to 2, no, it's for one singular glucose. So for one turn and all that we did, for only one of them, one of the pyruvates, or what you can say one of the acetyl-CoA, it's gonna be 36 to 38. It's not gonna be for both of the glucose, okay? And also, what else do we have? The anaerobic respiration. I'm just gonna look at my notes for a moment. Just give me a moment. Oh yeah, okay. A little note also. There's no photosystem in the cellular respiration. Okay, yeah, there's no photosystem in the cellular respiration, okay? Yeah, okay. So now we're gonna go to the anaerobic respiration. What was I saying? Some cells can function at a short time when oxygen is low. Some prokaryotics are anaerobic organisms. They live without oxygen, and in some cases, these cells continue to produce ATP through glycolysis. Okay, give me a moment. Ow! What did I just do? However, there are problems with solely trying to rely on glycolysis for energy. Glycolysis provides only 2 net ATP for each molecule of glucose, and a cell has a limited amount of NAD+. Glycolysis stops when NAD+ is used up, and the anaerobic pathway that follows glycolysis is anaerobic respiration or fermentation. The fermentation occurs in the cytoplasm and regenerates the cell supply of NAD+, while producing a small amount of ATP. Here are 2 types of fermentation. Oh, I heard about this part. Wait. I gotta check where I got my downloads. I downloaded this one. I was gonna open in time. Okay. We're just gonna have to wait a while. We're gonna also like go through a lot now. But this is like kind of the last part, you know? And I don't think so it's gonna be that hard, is it? Okay. Where did we stop? Health connection, lactic acid fermentation. Oh yeah, this is the part where I was talking about earlier, the one in the fermentation for the lactic acid and alcohol and carbon dioxide. Okay, so where's that PowerPoint? Where did it go? Okay, you know what? Wait. Let's just not think about it too much. So here we go. In lactic acid fermentation, enzymes convert the pyruvate made during the glycolysis to lactic acid, as shown in Figure 14. Yeah, that involves the transfer of high-energy electrons and protons from NADH. Skeletal muscles produce lactic acid when the body cannot supply enough oxygen, such as during periods of strenuous exercise. Oh, okay, I read that wrong for a moment. When lactic acid concentration in the muscle increases, the muscle becomes fatigued and sore. I said that already. That's cramps, by the way. What? Huh. Okay, where is the part of that? Okay, where is this part? Yeah, so here's the glycolysis part. Yeah, so the glucose that produces 2 ATP and has pyruvic acid. There's gonna be two versions. One is anaerobic and one is aerobic. The one that's aerobic is gonna be the one with oxygen present. That's gonna be the Krebs cycle, or ETC, known as electron transport chain, also oxidative phosphorylation. So the one that's anaerobic, the one that doesn't need oxygen, is gonna be fermentation, and the one with no oxygen gonna produce lactic acid, that's for animals, and ethanol, that's yeast. So here's a little like a little photo or graph or bar, whatever you want to know. So as we said, for the aerobic that needs it, it occurs in the mitochondria, and the glycolysis will occur. The ATP yield, or the net, or the whole thing that we're gonna be getting in total, is gonna be 36 to 38. Okay, so the glucose will be completely broken down, and the end product is water and CO2. As we said, after the Krebs cycle, there's gonna be a lot of CO2 released, so this is what is supposed to be expected. In anaerobic, we have in animals as muscle cells, or even for us humans, and we will not require oxygen. It occurs in the cytoplasm, which is the liquid of the cell. Okay, what am I saying now? Oh yeah, it is in the cytoplasm. Why am I doing this? So for all of them, glycolysis occurs. So glucose will be broken down. For here we have 2 net, and it's about 2 to 4 ATP. But on the anaerobic one, the glucose will not be produced completely, broken down completely. What am I even saying at this point? So it's not gonna be broken down completely, and you're gonna produce lactic acid. In the yeast, you're also not gonna require oxygen. It's gonna also happen in the cytoplasm. Your glycolysis will occur, and it's gonna be the same majority like the animal and muscle cells, but the thing is, your end products is gonna be ethanol and carbon dioxide. Now let's go look. We have other things to do. Yeah. Okay, you know what? We're just gonna joke. So now we're gonna get to here. We're gonna look at this. So this is the glucose. That's glycolysis. You get the 2 pyruvate, and after that you produce 2 lactic acids. So this one, the one that produces lactic acid, is gonna be for us humans and animals and the muscle cells, you name it. But when you get to the glycolysis here on the right, which is the alcohol fermentation, which we talked about, is gonna be the 2 pyruvate produced. You're gonna get 2 CO2. You're gonna get 2 acetaldehyde. I would not be concerned if someone's slapping right now for this pronunciation. I'm sorry. And you're gonna produce 2 ethanol afterwards. So now the alcohol fermentation, we're gonna read this out loud. It occurs in yeast and some bacteria. In figure 14, it shows the chemical reaction that occurs during alcohol fermentation when pyruvate is converted to ethyl alcohol and carbon dioxide. Similar to lactic acid fermentation, NADH donates electrons during this reaction and NAD plus is regenerated. So, science usage versus common usage. Okay. In science, this is relative to amount of substance dissolved in another substance. The concentration of hydrogen ions is greater on one side of the membrane than the other. In common usage, the directing of close undivided attention, the student's concentration was focused on the exam. Oh, it's the word concentration. I was wondering what they were talking about. Okay, we got this. So now in the photosynthesis and cellular respiration. Is this like a difference? Oh yeah, okay, since I'm probably guessing someone already studied photosynthesis, but the photosynthesis is the one that happens in the chloroplast that you're gonna see here. And on the right is the mitochondrion, and that's the one I'm learning about today. The one on left is a different subject completely. I might actually try to live that later. But seeing how I explain, there has some doubts. Yeah, but that can be fixed, right? So the photosynthesis and cellular respiration, including the anaerobic processes, provide the most energy for life processes, as shown in figure 15. Shows the interrelated cyclical nature of photosynthesis and cellular respiration. The products of photosynthesis are oxygen and glucose, and the reactants needed for cellular respiration. The products of cellular respiration, which are carbon dioxide and water, are the reactants for photosynthesis. You guys see that? Okay. Well, we're done. That's crazy. Okay, I kind of forgot to say an outro. I don't have an outro, but I'm just saying that I'm kind of glad I got to finish the cellular respiration lesson. Other than that, take care. If something was unclear, I have to recheck the whole video and try to correct myself. Either way, thank you.